So these are the fermenters which I was talking about even in the last class, that on page number 182 of your NCERT, fermenters or big stainless steel containers, which are also called as bioreactors, also called as fermenters, are drawn. Now in these containers, it is pre-sterilized, so no microorganism or bad organism is there inside, and they are sterilized, and then there are system to maintain the pH system to understand the temperature, pressure, etc. So there is one point for the material to be added inside and the other point for the exit. So inside these, when microorganisms are added along with their nutritive medium, they start preparing products of our own interest. So this is the importance of fermenter. We'll be learning about fermenter in detail when we'll be doing the biotechnology chapter, that means the next unit. So after fermentation beverages, we even discussed the topic of antibiotics in the last class. Anti means against the life, biotics means, bio means life. So microorganisms, there are certain microorganisms which prepare biochemicals which work against the life of the other microorganism. And when we say other organism, basically these other microorganisms are disease causing or harmful type of microorganism from the point of human beings. So that means antibiotics are against the life of microorganism and they are for the life of the human beings. So antibiotics are chemicals which are produced by some microbes and they kill or retard the growth of other, that means disease causing microorganism. We learned in the last class that the first antibiotic discovered was the discovery of this antibiotic was a chance discovery, and I told you the word in the last class, serendipity for chance discovery. And this antibiotic is penicillin, which was discovered by Alexander Fleming while performing experiment on Staphylococcus aureus, and he found Penicillium notatum growing on some of his plates, and I told you that he established the importance of antibiotics, but the real potential of the antibiotics was first established by Ernest Chain, Howard Florey. That's why NCERT clearly states that these three scientists, that means Fleming, Chain and Florey, three of them were awarded Nobel Prize. They shared Nobel Prize in 1945 for this marvelous discovery that how it can be used to save life of human being. Now, we need to remember that penicillin is the first one, and it is from the yeast called fungi called as the Penicillium notatum. But please remember, it is not just the only species. There is Penicillium chrysogenum also, which also synthesizes penicillin. But apart from the fungus, there are some bacteria which are called as the actinomycetes, and the species, the genus basically is Streptomyces. So we learned in the last class regarding this also, that there are bacteria and then there are actinomycetes which also prepares and synthesize good amount of various diversity of antibiotics. So we learned that bacteriacin comes from Bacillus subtilis, polymyxin comes from Polymixa, Bacillus polymixa, and then there are huge array like chloramphenicol, tetracycline, erythromycin, terramycin. So there are various types of antibiotics which we today obtain from various microorganisms. And we learned that how antibiotics function. They function by stopping the process basically of translation. Otherwise, there are some antibiotics which disturbs the plasma membrane or disturbs the cell wall. So these are the various methods antibiotics function. And then we learned that a good antibiotic should be broad spectrum, should be harmless to the host organism. That means in this case we are talking about human being, that it should be harmless to human. But we all know that today science understands that antibiotics do cause problem to the host body also, and it should not have any harmful effect on the good microorganism inside the gut of the host organism. And today we know it causes problem even to the microorganism in our gut. But one good thing is there, that it should be quick in action. We learned in the last class that there are broad spectrum and there are narrow spectrum type of antibiotics. Now today we are going to start with the next topic, and that is regarding the chemicals, enzymes and other bioactive molecules which are obtained by the microorganism. So there are chemicals which are called otherwise as the organic acids. For example, acetic acid, which is also called in local language as the vinegar, is obtained from Acetobacter aceti. Then we have the lactic acid, which comes from Streptococcus lacti, Lactobacillus bulgaricus. Citric acid, which comes from Aspergillus niger, Mucor species, Candida lipolytica. Then we have gluconic acid, which comes from Aspergillus niger, Penicillium various species, and butyric acid. Butyric acid is the acid which is found in the butter. Butyric acid is obtained from Clostridium butylicum. So when you read the same topic from the NCERT, it gives you the almost similar type of names, and remember, these names you have to by heart or learn properly for your board exam, because from these chapters straightforward question answers come with examples. Now apart from this, we all know that yeast gives us alcohol. So there are microorganisms which give us alcohol. We know Saccharomyces cerevisiae. And then there are microorganisms which even give us enzymes. So there are some examples which I have mentioned here. Enzymes which are also called as the biocatalysts.